Hello viewers, welcome to our channel. Today we're exploring an incredible medical breakthrough, how gene editing therapy was used to save a baby's life. We'll break down what gene editing is, what CPS1 deficiency means, why ammonia is dangerous, and how the amazing CRISPR technology works, all in simple terms. Let's get started. Recently, doctors in the US treated a baby born with a rare and life-threatening genetic disorder called CPS1 deficiency. But what exactly does that mean? CPS1 deficiency is a condition where the liver is missing a special enzyme called CPS1. This enzyme is super important because it helps turn ammonia, a toxic waste product, into something our bodies can safely get rid of. Normally when we eat protein, our bodies break it down, and one of the leftovers is ammonia. Ammonia is dangerous because it can damage our brain and other organs if it builds up. Here's how it works. The liver uses the CPS1 enzyme to start a process called the urea cycle. This cycle changes ammonia into urea, which is much safer. Urea then travels to the kidneys and leaves our body when we pee. But in CPS1 deficiency, this process doesn't work, so ammonia stays in the blood and can quickly become deadly, especially for babies. So how did doctors help this baby? They turned to a powerful tool called gene editing. Gene editing means changing the DNA instructions in our cells to fix problems. Think of DNA like a recipe book. If there's a typo in the recipe, you might end up with a cake that doesn't rise. Gene editing lets scientists correct those typos. One of the most exciting gene editing tools is called CRISPR. CRISPR acts like a pair of tiny programmable scissors. Scientists use it to find the exact spot in the DNA where the mistake is. Then, a special tool called a base editor can change just one letter in the DNA code, fixing the error without cutting the whole strand. For this baby, doctors designed a custom gene editing treatment. They delivered it through an infusion, where tiny particles carried the gene editing tools straight to the liver cells. There, the faulty gene was corrected, allowing the baby's body to process ammonia safely. After treatment, the baby started to thrive. His body could now handle ammonia, reducing the risk of serious damage. He'll need regular checkups, but this therapy has given him and his family new hope. Gene editing is opening up a new world of possibilities for treating genetic diseases. Thanks to science, what once seemed impossible is now changing lives. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe for more easy-to-understand science stories. See you next time.